Hey, AP Bio. So we're going to take a look at this topic 2, free response question A. So we can kind of go over this bit with you guys. This is the type of question that you're going to see on the AP test. We've been kind of prepping you guys for this the last 2 weeks. The two questions, one of them is a 10-point question. The other one is going to be a 4-point question, it looks like. And again, no graphs, no diagrams. You do not need to upload anything. All of your responses can be typewritten. The way it looks now, you'll be able to type everything right into the screen that you're taking the test on. And you don't need to use a lockdown browser either. So here's most likely an example of something you'd see where you're going to be asked to talk about different concepts, different biological processes, and then make some predictions. So this one was taken from an old AP test back in a while ago. But anyway, I like this one because it does address some of the things with cells. So, okay, during an investigation of a freshwater lake, we have the student discovering this unknown microscopic organism. So, what are 3 membrane-bound organelles that should be present in this organism? Describe the function of each. So everybody did really great with this. The only issue is a bunch of you guys selected ribosomes as one of the membrane-bound organelles. Ribosomes are not membrane-bound. Please remember that. Your membrane-bound organelles include things that have a physical lipid membrane around them. Okay, and so what would those look like? Well, let's take a look. So these are your membrane-bound organelles. Okay, you've got the nucleus. We're not going to say ribosome. Okay, this had asked for just organelles. I wanted membrane-bound. So it's nucleus, ER, which can include your rough ER, smooth ER, mitochondria, chloroplasts, vacuoles. Okay, Golgi bodies, lysosomes, and peroxisomes. Okay, those are your membrane-bound organelles. Let me highlight these for you. I believe I have the ability to do so. Yep. So these are your membrane-bound organelles. Okay, and their functions. Okay, that's what you wanted to talk about, and that's where you would get your points. Okay, we don't want cilia and flagella. Okay, and so that's the first part. And you had to mention any 3 of these. Okay, this has 4. We modified it a bit. Second part of the question asks you to describe 2 organelles that are in the eukaryote and how a prokaryote carries out the function. Okay, and so if you mention the nucleus, prokaryotes, that information's in their cytosol, it's in their cytoplasm. For endoplasmic reticulum, instead of using this to move materials around, they simply have materials diffused in their cytosol. Okay. If we look at the rough ER and the smooth ER, the same thing. Everything's happening in the cytoplasm. Okay. If you mention mitochondria or chloroplasts, they're going to use membranes. Okay. They're going to use their cell membranes to carry out cell respiration and photosynthesis. Okay. Remember, these prokaryotes, okay, they don't have membrane-bound organelles. So they're simply going to use their cell membrane in order to properly carry out the processes of cell respiration and photosynthesis. Okay, and if you mention a vacuole or a vesicle, they use large molecules or they have granules. There's things called inclusion bodies, which are basically material that's been taken into the cell that's included within the cytoplasm. Okay, so you needed to mention any 2 out of the 3 that you talked about. Okay, now if you mention ribosomes, we didn't double jeopardy you, so you didn't lose the points in part B if you just talked about the ribosomes in part A. Okay. All right, moving on, it asks you to talk about the endosymbiotic theory. Okay, describe 3 observations that support the theory of endosymbiosis. Okay, any 3. Really, the ones that we talked about are a bunch of these. Okay, mitochondria and chloroplasts have their own DNA. They can self-replicate. They're circular. They contain ribosomes. They have a double membrane. Okay, having that double membrane is similar to the membrane of prokaryotes. Okay, so any of these bullets that we just highlighted would definitely take care of that. There are other things here too that we've never gotten into. Oh, here, the size of bacteria. They use a lot of enzymes similar to prokaryotes. That's fair game too. Okay, so pretty much as long as you mentioned instead of 3, we had you just mention a few observations. That was it. So 2 observations that support this. So there you go. They have their own DNA. They self-replicate. They have double membranes, okay, separate enzymes. Okay, the advantage of eukaryotic cells containing multiple organelles. Having multiple organelles means compartmentalization, okay? So that increases efficiency. That's a key thing, okay? By compartmentalizing and separating these reactions into different locations, now you can specialize the reactions and they become highly efficient, okay? So instead of being a jack of all trades, master of none, when you compartmentalize, now each unit within the cell becomes highly specialized, more efficient, and that leads to better productivity for the cell and increased chances for survival, okay? All right, for the last one, surface area to volume ratio. Okay, they gave you two cells. Cell A had 32 micrometers squared surface area and a volume of 8 cubic micrometers. And cell B was a little bit bigger, 36 and 12. So they wanted you to calculate the ratio of the surface area to volume for the cell that is predicted to exchange materials with the surrounding environment more efficiently. That would be cell A. Why is that? Cell A is smaller. Remember, a smaller cell always has a higher surface area to volume ratio. So if you calculated that, you would see that that ratio is 4, okay? All you do is divide the surface area by the volume, and then the number you get is a whole number, and so that ratio is 4. The larger surface area provides greater efficiency, okay? That's going to help maintain a smaller volume, okay? So the reason why it's more efficient is because the larger the surface area is to the volume, the more efficient the cell is at transporting materials in, okay? All right, let's go on to question B. Question B here says ribosomes. We find ribosomes in the cytosol as well as attached to the ER. Okay, and so we know that the proteins on the attached ribosomes are delivered to the ER, and the other ones go to the cytosol. So explain the ultimate destination of a protein on an attached ribosome. Okay, so you don't have to talk about the function, just where does it go? So if it's attached, it's going to stay within, if it's attached, it's going into the ER, which means it's enclosed in a membrane. From there, it's going to go into a vesicle and then possibly to the Golgi, and then from there, it gets exported out of the cell. So for letter A, the destination of a protein produced on an attached ribosome could be that it goes to the membrane, or it is set up for secretion, excretion, or transport out of the cell. Okay, what can that protein do? That protein can be a receptor, it can be a hormone, it can be a signaling molecule, it could be a docking protein that's on the outside of the cell, it could be something that's used for attachment on the outside or inside of the cell membrane. Okay, and a protein produced on a free ribosome, it's going to be within the cytosol, so that's going to stay in the cell. All right, so proteins made on free-floating ribosomes in the cytoplasm or in the cytosol, they remain inside the cell. Those are proteins that can be used for enzymes, digestion within the cell. Those might be utilized in the nucleus, so they might be involved as transcription factors. These could be things like DNA and RNA polymerases. Okay, these might be enzymes involved in cell respiration. Okay, enzymes involved in glycolysis. Okay. All right, and that's it. And again, for this question, you got 2 points for each part, so a total of 4. And that's it. Okay, so we kind of started you guys off a little easy on this one, hopefully, just to kind of get a little recap of the organelles and some of their structures and functions and what they do. And that's it. Okay, so I hope that worked well for you guys. Again, shoot us an email, give us a remind if you have any questions, and we'll see you online. Thanks, guys. Have a great day.